Tomorrow, bibliography, annotated bibliography is due. So I already got a couple of them, haven't looked at them yet, but you can just put them into Blackboard in the Dropbox, and then bring those Guess My Hypothesis worksheets with you tomorrow. We'll continue to work on those during lab time. So, lots of consistent issues with the homework for this particular section. So I have built in some time for us to work on that and to talk about how do we actually take DNA, convert it to RNA, and then ultimately translate it into an amino acid sequence. So, take them both. Share with a friend. Today's main goal is to talk about this flow of information, from DNA to RNA to protein. And this becomes really important, or where we're going to come back to this is when we start to talk about genetic mutations. Again, we're going to talk about genetic mutations primarily in the context of antibiotic resistance in the next couple of weeks. So know that what we're talking about today is the molecular basis for antibiotic resistance. Okay? Let's start out with a multiple choice question. All right, so the question reads, what is the correct sequence of RNA made from the following template DNA? And the 5' end of DNA reads ACCTAG, and then the 3' end. So there are 4 options out there. This is not one that you can solve in like a half a second, so I'm going to give you time to look at the answers, strategize a way to come up with the correct answer for this one, and then talk it over with a partner. I'll give you about 2 minutes to do this, and then we'll discuss it. Go. They all look the same, yes. Yeah. That's why, what could be a strategy for this one?
I went for the ones that started with 3'.
That's one way you could do it. Okay. What else could you do? Would you be able to solve this on your own page? Without even looking at it? Like if this were in a multiple choice question, but it asked you just to fill out the answer. Try that. Write it out on your own and see which one best fits with what you've put. What are key characteristics of RNA that make it distinct from DNA? What's the most basic one that we oftentimes talk about? Use and T-switch, right? So instead of thymine and DNA, we find uracil in RNA. There's other differences with respect to the sugar base in it. We're not going to really worry about that right now. All of these have use, so that's not an issue here. What are other features that we're looking for between template DNA and mRNA? 2 key characteristics. Anti-parallel, complementary. So we need to make sure that both of those things fit. We need to make sure that there's complementary sequence and that complementary sequence is in the right polarity, basically. Before even answering this, what strategies are you using to try and identify the correct answer? No one has a strategy. Maybe we'll make this the first question on the next exam. Well, what was your strategy? That's okay. I didn't ask if it was right or wrong. I just asked, what was your strategy?
Okay, I guess this is what I get confused because I thought, like, looking at the slides in the lecture, and this one was 5 prime. So I just like in my own, I did the 3 prime to 5 prime to the complementary DNA,
Okay.
and then switched the T's and U's out for the mRNA. But I got one that's not.
So if I'm understanding what you said correctly, you wrote it yourself or something like that. And that's the strategy I'd use. I wouldn't even start by looking at the multiple choice. I'd probably write this out. So if we were, what did you have, Molly?
TGGA2C.
Okay, TGGATC, but we'll swap out the U's or the T's for U's, right? So U, G, G, A, U, C, 5 prime. And you're right. That, I'm pretty, actually, it does show up there. Which one?
It's the first one. Sorry, I mean obviously I can read.
Well, it's not the first one. It's actually the second one here, right? Because that's the one that has our. So it's there. And if you were to fill this out in an exam, if you wrote it down but you somehow answered the wrong multiple choice question, I'd actually give you the credit for it because you've shown me that you can do it. All right. I think the
mix up was the 5 prime 3 prime.
So yeah, and that's a key thing. I think we always want to make sure that we include those in there because that tells us about the direction that we're reading this information as well. So one of the common issues with the homework assignment was not listing what is the 5 prime and what is the 3 prime end of the mRNA. And again, in relationship to the template DNA, the mRNA is always anti-parallel, okay? And always complementary as well. So the first thing is label those 5 prime and 3 prime ends so that you understand which direction the information is being read in. Any other strategies that folks tried to use on this one? Okay, I think that might be the best, is to write it out on your own before trying to solve the problem on your own. All right. Let's, here's what I want to do. I actually want to go to the homework question now. And I'm just going to go up to Blackboard and post it. Not Blackboard. Okay. So again, what this question is saying, and you're right, the DNA here is written in a different direction than the DNA that we looked at in the last problem. And to be honest, we write DNA in both directions all the time. And that's where those polar ends, the 5 prime and 3 prime end, help us to decide the direction. Okay? In this case here, the template is going to be polymerized, or the RNA is going to be made in the 5 to 3, and that means that the polymerase is moving in this direction. So adding U, C, U, A, C, C, so on and so forth, all the way across to make that complementary RNA sequence. What are we looking for in an RNA? If we want to take an RNA and translate it into a protein sequence, where does translation begin? So what's that location on or what's that location called where translation actually begins? Alexi? Start codon. What is the start codon? There's only one. AUG. So the first thing we need to look for here is we can't just start at the beginning of the mRNA sequence. In fact, in all mRNAs, eukaryotes and prokaryotes, there's this region that we call the 5' untranslated region. It comes before the start codon. So we need to identify here, where are there potential start codons in the problem? In other words, where can we find an AUG? Okay? So what I want you to do I think I'll write this out real quick and double check your answers as I'm writing this out, and then we'll go ahead and we'll scan for that first AUG. All right, so this will be my 5' end, and U, C, U, A, C, C, A, U, G, G, U, G, A, G, A. Oh gosh. G, G, C, C. Can I just stop? Is that okay right now? Yes, I get to say that it's okay. All right, somewhere in there is our start codon. Where is it? Identify it for yourself. And if you've identified it correctly, it should be, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9th, and 10th nucleotide in this mRNA sequence is our start codon. Okay? So some folks were taking this mRNA sequence and translating it right from the beginning. Instead, what we need to do is we need to identify where does translation actually begin from, and that's the AUG. Not only does the AUG encode for start, but it also encodes for the amino acid methionine. So that's the first amino acid that's going to be placed in there. And then what do we do? How do we read this afterwards to determine the amino acid sequence? So how do we actually read to determine the amino acid sequence? Some folks actually did this right. Not that I'm surprised by that. That came off wrong. Some people did this right. About half of everyone was able to do this problem all the way through with just very minor mistakes in reading the amino acid sequence. Here's what we'll do. I want you to talk to your partner for 2 minutes and come up with a strategy on how do you go from this start codon and now convert this to a protein or an amino acid sequence. Okay? So talk it out with someone else. Including our visitors today as well. Don't let them feel alone. Yeah.
Yeah.
This mRNA here, this is our start codon. How do we go from that and figure out our amino acid sequence? Alexi. And what's every group called? What term do we give to that? Do you know, Allison? No. Oh, you don't get... I know somebody said it, but shall we play a quick game of Hangman? Oh, dang it. This is the only way that gets you guys to talk. Okay, codon. So every 3 is known as a codon. All right? And so Lexi's absolutely right. Group them in 3s. There are strategies to do this. And the way that I do it, if I'm doing this on the computer, I'll put a space in between each 3 so that it's easier for me to read. If you're doing this on paper, the way that I'll do it is I will just draw lines in between each codon. Oh, shoot, that's not one. Oh, no. Oh. You get the idea. Okay. So we would group them into 3s, and then each 3 corresponds to what? Amino acid. Okay, so for example, AUG is methionine. GUG is what? And I wouldn't expect you to know this off the top of your head. Actually, that would be painful because there's 64 of these. So I've given you guys a worksheet that has the codon code on it, and when it comes to exams or anything like that, I'll give you one of these to use. Okay? So if we look at this codon code here, GUG encodes for what 3 letters? VAL, which is valine. Okay? And then the next one on the list is AGA, which encodes for which amino acid? Arginine, and so on and so forth. We say that this codon code is degenerate. And what we mean by that is that there are certain amino acids that are encoded for by more than one codon. So for example, let's look back at valine real quick, GUG. What else encodes for valine? What other codons encode for valine? What was that, Katie? GUU, GUC, and GUA. So in total there are 4 there. Note that in each of these, or at least in most of the cases, what the nucleotide of the codon that's changing is that third one. Okay, so, and these could represent different codons for that amino acid, or as we're going to transition into in the next couple of minutes, if we have mutations. If there's a mutation of a GUG to a GUA, it's what we call a silent mutation, one that has no effect on the amino acid sequence. Okay? And this is in contrast to missense and nonsense mutations. All right. So reading this sequence, at what point do we stop? At what point do we stop reading or translating an amino acid sequence from an mRNA? What are we looking for? Stop codon. And there are 3 of them, right? University of Georgia, UGA. Okay. University of Anchorage, Alaska, UAA. And then some South American university that I can never remember the name of because we don't have a UAG in the United States as far as I'm aware. Okay, I remember these in terms of colleges, but UGA, UAG, UAA are the 3 major stop codons. What we're looking for is what we call an in-frame stop codon. One that when we're reading in those sequences of 3 will produce one of these 3. An out-of-frame stop codon does not stop the process of translation. And so if you're trying to identify what we call an open reading frame, region that encodes for a protein or an amino acid sequence, start with the start codon and then try to identify an in-frame stop codon. If you can find that, that's considered to be an open reading frame, or considered to encode for a protein. Okay? Let me check one more thing real quick before we move on. We'll come back to that if we need to. All right. Another multiple choice question. Using your codon codes, and I close this bad out, so. Okay. Which of the following is an example of a silent mutation? So we're going to switch focus for a couple of minutes and talk about mutations now. So is it A, AUG to UUG, B, UGG to UAG, C, AAG to AAA, or D, AGG to ACG? Use your codon codes, identify the best option. I'll give you a minute, and then double check with someone else. What's the best option? A, B, C, or D? Taylor? Come on.
C.
Why do you say C?
Because they code for the same thing.
Perfect. Perfect. Silent mutations encode for the same exact amino acid both before and after the mutation. What do these other ones represent? So the first one there, we're going from an AUG to a UUG. AUG encodes for methionine. What does UUG encode for?
Leucine.
Leucine? So this would be an example of what type of mutation? Yeah, this is a missense. A missense mutation is one in which the amino acid before and after are different from one another. Okay. What does B represent? So what does UGG encode for? Tryptophan? Okay. And how about UAG? Jennifer, do you know? Yeah, it's a stop codon. So the term that we give to this one is a nonsense mutation. Sometimes these stop codons are called nonsense codons. The reason why we give that is because they don't sense for any amino acid. That's the reason why we call them that. So three major types of what we call point mutations, where one nucleotide is being exchanged for another. Those three types would be the silent, no change in amino acid sequence, missense, changes the amino acid, and then nonsense, changes the amino acid to a stop codon. And so therefore it would stop the translation of the protein early. Out of the three of these, we consider the stop codon most frequently to be quote-unquote most harmful to the organism because of the fact that it doesn't produce the full-length protein. Now there are other types of mutations beyond these. There's insertions and there's deletions as well, in which an extra nucleotide gets added or a nucleotide gets removed. What does this do to the amino acid sequence? So if, for example, we had a codon sequence like this, where we had AUG, CUC, ACG, and we added, or not we, but the DNA polymerase, our RNA polymerase has made a mistake in the genome, therefore changing the mRNA sequence to AUG, CUU, CAC. What does this do to the codon code, or the reading frame? It moves the whole thing. So what does that do to every single one of these codon? It changes these codons downstream, which means what about the amino acid sequence? Exactly. Changes every single amino acid downstream of that. So these insertions and deletions are actually can be much more harmful to the organism than can even these point mutations, where we're just changing a single nucleotide. Okay? All of these mutations arise as mistakes of DNA polymerase. DNA polymerase is really good at replication, but it does make mistakes. Just to kind of go over the statistics of it real quick. Let's see if we can get out of here. There we go. So statistics on DNA polymerase. Makes a mistake 1 out of every 10 to the 9th or 10 to the 10th base pairs that it replicates. That means in the human genome, it's making a mistake maybe 6 times per replication, but most of these mistakes get corrected by additional repair mechanisms that we're not going to have time to talk about. In the bacterial genome, it makes a mistake maybe once every 200 to 2,000 generations. So within a culture, you can get maybe 10, 12 different genetic variants within a test tube growing the cells overnight. We can increase the rate at which DNA polymerase makes mistakes, and that's done with what we call mutagens. Things that will disrupt the DNA polymerase or disrupt the DNA itself. What are examples of mutagens? Some of these we actually use to control microbial growth. Some of you may have even been exposed to these over spring break. Radiation of what form? UV, all right? So UV radiation is a strong mutagen. What it does is it takes adjacent, if you have, for example, a sequence of DNA that has two thymines next to it, what UV does is it actually creates a base pair between those two thymines. And it's not on the backbone, it's actually between the DNA base itself of those two thymines. So it creates in the DNA sequence actually a little bit of a pucker. Instead of having perfectly linear DNA or helical DNA, it takes it to a format where there's like this little bend. If that doesn't get recognized by host repair machineries, that sequence of TT gets misread by the DNA, or by the DNA polymerase, leading to potentially insertion, deletion, or just a point mutation of misreading that sequence. Okay. Other mutagens, ionizing radiation, ionizing radiation or X-ray. That's why those are two really good controls of microbial growth, is because mutation of the DNA is going to stall or inhibit the growth of the microorganism any further. Microorganism has to repair that DNA in order to divide again. And if we provide enough UV light or if we provide enough ionizing energy, what that does is inhibits, or damages the DNA beyond repair. And so the cell just kind of stalls its replication and lives out the rest of its life in a quiescent or quiet state. Questions on mutations or DNA to RNA sequence? Okay. I think what we'll do for the rest of the time today then is work on this worksheet for a while, because I think this will help to make a couple of additional points that I wanted to. This one is taken out of a textbook that I've used in the past. And there's questions A through I on this one. We've actually covered all the basic information to answer all of these now. So what the problem is going to ask you to do is to first figure out the DNA sequence given the mRNA sequence and the tRNA sequence. So one of the things that we have to understand is what is the relationship between tRNA and mRNA, and then between the mRNA and the template DNA. So that'll be one of the first things to address. And then you have to figure out the amino acid sequence based on the mRNA sequence as well. One good practice would be to write out not only the DNA sequence, but write out the full mRNA sequence, and also write out those tRNA anticodons. Okay? That is a good practice to work with here. And then it's going to ask you about different types of mutations and what would the effect of those mutations be on the DNA sequence. Right? So feel free to work with a partner or two partners on this one. I'll be kind of roaming around to answer questions as they come up. And maybe with about 5 minutes left, we'll at least go over the DNA sequence to make sure we've got that correct. All right, go. Yes, Molly. 8 minutes or so, 7 minutes. I want to make sure I mention homework for Friday. So this one tests a lot. This question is really testing what are the relationships between different molecules involved in this process of translation. Okay. So again, just to review, what is the relationship between template DNA, which would be this top one here, and the mRNA? What's true about their relationship? Two common features. They're complementary and anti-parallel. In other words, what end would we need to put on this mRNA sequence? This is our 5' end. So this allows us to very easily read the mRNA from left to right to figure out the codons. And then, of course, the other part is, as Molly said, they are complementary to each other. What's the relationship between a tRNA and an mRNA? Complementary. And they're actually anti-parallel as well. What is the function of the tRNA? Let's go back to Monday. So what does the tRNA actually do in the process of translation? What does it do? Transfers what? Go with it.
Transfers one codon or amino acid to the next.
Okay, so it's involved in amino acids. The tRNA brings the amino acid to the ribosome. And the tRNA matches its anticodon with the mRNA codon to bring the right amino acid to the mRNA sequence. Okay? If the mRNA and the tRNA bind together, just like DNA would to itself, it also means that the tRNA and the mRNA, these two have to be anti-parallel and they need to be complementary. So for example, complementary. So for example, if our tRNA sequence is UGG, this would be 3', this would be 5'. What is our mRNA sequence? ACC. Atlantic Coast Conference. What's our DNA then in those 3 locations? TGG. Note here, one thing to point out. Anybody know the old phrase, the enemy of my enemy is my friend? No? Apparently I'm old. Okay. Some of the high schoolers do, but that's okay. You do? All right. So enemy of my enemy, or UGG's relationship to DNA, tRNA and DNA and template DNA actually have the same exact sequence. So this anticodon here has that same sequence as that template DNA. Okay. Let's go ahead and let's finish filling out the DNA sequence here. Methionine, our first amino acid. What mRNA sequence encodes for methionine? AUG. What is our DNA sequence then? Very good. TAC. mRNA sequence, we'll have AAA there. CGU. What's the DNA, the corresponding template DNA sequence? Very good page. GCA. We've already said that for this one, it's TCC. And then for the last 3 nucleotides? Allison? For this one right here, these last 3?
No, the one before.
Oh, yes. No, wait. Yes, you're right. I apologize. Didn't you see these tails on those C's? You're right. Thank you. ACT as our last three there. Okay. Okay, mRNA to protein. AAA encodes for? Lyse. CGU? ARG. A pirate's favorite protein or amino acid. ACC? Trip. UGA? Stop. Also known as a nonsense codon. So question I asked for which of these is a nonsense sequence, it's that UGA there. Okay. Before I stop, I suppose I want to check, are there any questions with the mutation questions on this one? Any of the insertions or changing one amino acid to another? Okay, keep practicing with those. We're going to apply them. We're going to come back to that in about a week or so. Now, before you head out, just pay attention for 1 second. Homework for Friday's class. We're going into lac operon. So we're going to transition briefly to talk about how do genes actually get turned on or turned off. We're going to use one example. It's a classic example in bacteria. The homework for this one is a group wiki. It's a two-parter though. Okay, so pay attention to that. Part 1, every member of the group has to fill out one of the rows in the table, and then the second part is after everybody has done that, and that's due at like 12 tomorrow. Everybody go back and put in one trend you see in the data. So analyze that, okay? And we'll use that as our starting point for Friday's conversation. Annie?
What is it? I didn't see it.
I will make sure that it's up as soon as you guys exit this classroom, which I think is right now. So. Have a great rest of the day. I will see you all on Thursday. If you did not pick up your exam from me yesterday, I do have them.
For the Friday homework, it's like almost never completed by the time I go to work at 3. They do it after midnight.
Yeah, that's all right.
Because I just work until after midnight, so it's like if it's not all done before 3, I can't do it.
That's perfectly fine.